pathology which we frequently use in NGS. Okay, so what is a base pair? This is the basic building block of double-stranded DNA. Base calling, these terms will be using very frequently. We will talk about these. Base calling is the process of assigning bases to the chromatogram P. Read, what is read? Continuous sequence produced by the sequencer. Okay, that is the read. Read length is the number of bases sequenced. So one platforms or one sequencer is different from other in terms of read length. कि जैसे पैक बायो का रीड लेंथ कितना है, नैनोपोर का रीड लेंथ कितना है. Okay, then coming to the coverage, how many times the particular basis or region is read. So that is the coverage. Contigs, once you get the reads, you have to align or the overlap the sequences, the segments of the reads, and then make the contigs, and then we assemble them and we make the scaffolds and we do downstream analysis. Okay, so these are the various NGS platforms available in the market. You can look at the Illumina sequencing, and then Ion Torrent is there, PacBio is there, Nanopore is there, and then different variants of these sequencing technologies are also there. And all the sequencing platforms differ in terms of their sequencing chemistry, okay, and then data output, okay, their read length, but there are 4 common things or the 4 common steps in all the NGS. What are those? Library preparation, then clonal amplification, library of what? If for doing genomics, so library of DNA, and for transcriptomics, library of, and then clonal amplification, because initially you are provided with very small amount of DNA, so you have to amplify it. Okay. And then sequencing. This is again, chemistry may differ, but this is the common step. And then the last and very important part is data analysis. Okay. So what are the 4 common steps? Library preparation, sequencing, and data analysis. Okay. So as I said, sample your input material, whether you are taking planning for genome sequencing or transcriptomics. So your input material DNA or RNA, the quality should be very, very good. Okay. How will you assess the quality of DNA or RNA? Nanodrop or spectrophotometer. Very good. So your then you have to prepare the library, okay, and then the post processes you have to perform. So this is the common steps in library preparation. Actually, this library preparation is are done by kit. Okay, there are many kits available in the market. Like we have a kit from NEB, then TrueSeq hai, Nextera. So as per the recommendation or the protocol is given, you have to just follow the protocol. Okay, apna kuch naya usme hai hi nahi. If they have given like you have to vortex for 15 seconds, then you have to on the timer and just you have to stick with the time. Okay, so this is the library preparation. So you started with, there are many steps, and like you have to do the fragmentation, then you have to do the end repairing. These all are the in between library preparation process. Okay, size selections are there, and there is one important thing, adapter ligation. So what we do, I'll tell in a very broader way that we have a larger DNA. So what we do, we broken down to small. कैसे आप करोगे? Small बनाओगे? So by means of shearing और फिर sonication कर सकते हो, ठीक है? और then adapter ligation. So this is the common process, and then once library is done, we go for the cluster generation, clonal amplification. I will tell these things in detail, and then we go for the sequencing run. So for library preparation, what we need, what we require is RNA or DNA. Okay. So this is the there is one numerical assessment number given for RNA and DNA. For example, if I am planning for transcriptome analysis and I have isolated RNA, okay, I have checked that RNA and gel and other on spectrophotometer, nanodrop, I have taken reading. Okay, so even you have to check the quality or the RIN number. So this is the numerical assessment recommended for any samples. For example, if you are having sample for transcriptome analysis, so what will be the recommended RIN? That should be at least 7. Okay, so RIN value, if your sample having RIN value 7, then only you can proceed for the library preparation. Otherwise, you have to repeat your extraction process again. Okay. And for this one, these are all other. This is the chip. So how we do this? How we get this RIN number? This is the machine called bioanalyzer. Okay, I will show you this machine. So these all are the chip. This is specific for DNA and this is for RNA. So you can see here gradient is there, ladder is there, and these are the number of wells. So in one well roughly 10 to 12 samples you can load. Okay, you can check the quality. So after loading your samples, this chip is placed here in the machine and it is attached with the monitor. So you will be getting result like this in bioanalyzer. Bioanalyzer किस लिए है? आपका RIN value पता करने के लिए, ठीक है? तो if your sample having good RIN more than 7, this is the sample having RIN value 10. So you can see the sharp peak here, but here you need to repeat the your extraction process. Okay. These are some of the results we recorded in our machine. So, and along with your this peak, you will be getting the gel bands also. Okay, in bioanalyzer. Then the next step is cluster generation or clonal amplification. Here you want to amplify your sequences or the template. Okay, so how we perform the clonal amplification? This is the flow cell. किसी ने देखा है flow cell? Okay, so there are 8 channels in this flow cells. Okay, and these flow cells are coated with millions of oligos. Okay, and I'll tell you the sequence during the library preparation. What we do, we add adapter molecule. Okay, so the sequence of adapters are complementary to the oligos present here. So that's how this cluster generation or clonal amplification happens. Okay, and this is the machine called CBOT. तो आपका जो भी यहाँ पे आपने रखा है samples, here you will be providing your DNTPs, polymerase, all the things are required for the amplification. So this is about the cluster generation. And what is multiplexing? Multiplexing नाम सुना है? What is multiplexing? I told you this is
multiplexing is a more different type of DNA.
Okay, I will now you can tell me.
To multiplex PCR.
So as this name indicates, multiplexing. So here what we do, for example, if I am having 5 samples. Okay, so here we use barcodes or the identifier for each samples. आपने सबकी लाइब्रेरी बना लिया, extract कर लिया, but when you are going to run all the samples in one channel, how you will identify this is your sample and this is my sample? Okay, so this multiplexing is simply pooling of many samples, okay, and you can run in the single lane. ठीक है. So it allows large number of library to be pooled and sequenced simultaneously during a single run on Illumina instrument. Okay, with multiplex sequencing, individual barcode sequences are added to each DNA fragments or the each samples. Did you get multiplexing? Okay. As I said, in one lane, more than depending on the samples, more than 10 or 12 samples can be loaded. Okay. अब मैंने एक लेन ले लिया कि कंपनी से कि हाँ मुझे एक लेन आप दे दो, ठीक है, उसका जो भी पेमेंट होगा आई विल गिव. तो आई हैव 2 samples, you are having 2 samples. So once I am going to pool, so this is just the identification of each samples. So you will have to add some barcode, okay, या फिर आप identifier बोलो, ठीक है, tag बोलो, उसको हम करके फिर सारे samples को you have to pool, okay, and then after sequencing you have to do the demultiplexing, okay, you will get the data and then आपका tags are there, so you can identify कि ये वाला sequence हमारा है, ये हमारा है, and then yes, so on the basis of barcoding you can then do the multiplexing. Okay, so these are the different other variants of Illumina, like we have mini-seq, mi-seq, next-seq, high-seq, 4000 version. So these all are different in terms of, you can see here in these variants one flow cells accommodated in the machine, but here in high-seq X दो flow cells आप रख सकते हो, तो you can see the output will be just double. Okay, so we have 1000 version, this is very basic one, and nowadays we are having see NovaSeq 5000 and 6000 in market. So here you can see 4 flow cells can be accommodated in machine. Okay, and now coming to this Illumina sequencing technology, how it works. So as I said, what happens, so we start with the genomic DNA, cDNA, okay, and then this DNA are broken into small fragments, and what we do, we add adapter molecule. Adapter will go and bind with both the terminals of this DNA. This is also called as process DNA. Okay, and we are having flow cells, and what is there on the surface of flow cells? Oligos are there, and as I said, those oligos, or you can say they are complementary to the adapter molecule. So once you will put your adapter ligated DNA onto the flow cells, because of the complementarity they will go and bind. Okay, and so after sometimes once you will add DNTPs, polymerase, everything. So what happens, this adjacent oligos, it act as a primer, and it will start adding the synthesis of other strand. So firstly it will form the structure like this, it will start bend over. Okay, so once it get bend over, it will form the structure like bridge, and then the adjacent nucleate this oligos, this act as a primer, and it will start the synthesis of the next strand on this bended stranded. So it will form the amplify the second strand. So this is also called as bridge amplification. Okay, so this is the characteristics of Illumina technology. And after some time, then again this double strand DNA get denatured, and then again the process repeated. So that's how we get the millions of the copy of the particular DNA. Okay, and what happens here, this is based on the sequencing biosynthesis. So principle is sequencing biosynthesis. What is sequencing biosynthesis? Means So it is opposite. While synthesizing the new strand, you are getting the information that which nucleotide are being incorporated. Okay, because here the nucleotides are labeled with 4 different colors. Okay, so for example, C is there in the template strand which you want to sequence, and if G is added there, so you will be getting yellow fluorescence. Laser is there, it will record. Okay, so all the 4 nucleotides, they are labeled with the different fluorescent tag. Okay, and the incorporation of every nucleotides releases fluorescence. Okay, and that will be recorded by the laser. Okay, once you will get the reads, so you have to align those reads and then you can do the downstream analysis. So next technology is Roche. This is also known as 454 sequencing. Why this is called 454 sequencing? What is 454 in RAS? So this 454 is the first commercial successful NGS system. Actually it came before Illumina, but in 2013 it was acquired. In 2007 it was acquired by Roche, but because of the non-competitiveness, this technology, the hardwares and everything, they were stopped. Okay, so here also it also works on the principle of sequencing biosynthesis. Have you heard about pyrosequencing? Pyrosequencing, किसी ने सुना है? So incorporation of every nucleotide releases pyrophosphate. Okay, and those pyrophosphates are utilized by the enzyme sulfurylase. Okay, which तुम बताओ. What is pyrosequencing? How many enzymes are used in pyrosequencing? What is the role of epirase?
कंटामिनेशन का चांस रिड्यूस कर देता है.
Sorry.
और कंटामिनेशन का चांस रिड्यूस कर देता है. फॉर एग्जांपल अगर हम कोई न्यूक्लियोटाइड ले लें, तो ल्यूसीफेरेज से ल्यूसीफेरिन निकलता है. देन वो हमें सनलाइट देता है, सॉरी हमें लाइट देता है कि हमारे पास ये पर्टिकुलर न्यूक्लियोटाइड है. देन उसके बाद हम उसको एपीआरए से ट्रीट करते हैं जिससे कि हमें फर्दर कोई कंटामिनेशन ना मिले, मतलब एक्यूरेट हो.
So this epirase is used for to degrade the unincorporated nucleotides. Okay, so this ATP is further utilized by luciferin, which luciferase enzyme, which convert luciferin into oxyluciferin and produces light, and the intensity of light is measured here. Okay, so and these are the different variants. So here also the library preparation, as I said, this is common for all the platforms. The process may be different. So here in case of Illumina, what is the sequencing unit? Flow cell. Okay, here in place of flow cells, what we use? Beads. Okay, so this bead is used here, and as in flow cell, the cluster generation occurs on the surface of flow cells. Okay, here it is occur on the bead. So here, and then normal PCRs, here it is emulsion PCR we used. Okay, and one picoteter plate is used, and this picoteter plate, there is millions of wells are there, and wells are designed in such a way that one bead can accommodate it in one well at a time. Okay, so just to know you, what is the application. Then coming to the ion torrent. Ion torrent का नाम सुना सबने? So this is principle is similar to pyrosequencing, but here in case of PPI, pyrosequencing, release of H plus is monitored. Okay, release of H plus causes changes in the pH, and one sensitive layer is there which actually record the change in the pH. Okay, so what happens here? You can see here this ion sensitive layer is there. So incorporation of nucleotides or the release of H plus causes change in the pH, okay, and this pH change will be monitored by this layer. So what happens here, the whole chip is flooded at a time. If you are adding DATP, so only one time DATP will be added, then you have to wash them and then you have to add another nucleotide, like that you have to do, okay. So the nucleotide flood the chip with one nucleotide after another. If the next nucleotide that flood the chip is not a match, no voltage change will be recorded. And if there are two identical bases, then what will happen? Voltage will be just double. Okay. And these are the variants of ion torrent. You can see here personal genome machine is there, then ion protons, and these are the different chips we use. Coming to the PacBio sequencing. So in PacBio, it is also known as SMRT, single molecule real time sequencing. Okay, this is this belongs to third generation sequencing platform, and this is the platform which generates longer read length. Okay, so the read length of PacBio is around 60 kb. Okay, and sequencing chemistry is slightly different here. So what we the major sequencing unit, or you can say the component, is this ZMW, zero mode web guide. Okay, that is the sequencing unit, and this is the template which you want to sequence. So after adding adapter molecule, it will go and bind at the both terminus and it will form the hairpin like structure. Okay, so this is the template is known as SMRT Bell in case of PacBio. Okay, and this is SMRT cell where sequencing takes place. So what happens in here, there are millions of ZMWs, okay, and on the base of the ZMW, polymerases are embedded. Okay, polymerase is there, so it will be like this. Okay, so you can see here polymerase in ZMW. So once you will put your this template to be sequenced here, and here also your nucleotides are labeled with the different fluorescence. So on the basis of fluorescence, it will be recorded. And there are two different variants in this PacBio RS2 and then sequel. Okay. Coming to the nanopore sequencing. nanopore सुना है? देखा? It is there in the discovery. Nanopore है ना sir?
Nanopore, we have an exclusive one day and some training as far as the demo that.
Yes sir, yes.
Day to day or night to day they have.
It is there in discovery center. You can have a video. Okay, so as the name indicates, nanopore sequencing means the DNA which you want to sequence has to pass through a pore. Okay, and the size, why this is nanopore sequencing, because the size of the pore is in nanometer. Okay, so it's roughly 100 to 200 nanometer pore size where DNA will be passed. Okay, so what happens here? So two proteins are there. One protein actually it will unwind this DNA helix. Okay, so that only one strand can pass here, and the second protein creates the pore in the membrane. Okay, and each bases, A, T, G, C, whatever bases are there, they produces different ionic current. Okay, with the different degree. A flow of ion through the pore creates a current, and each base blocks the flow to a different degree, and the degree in the changes in the current that will be monitored. Okay, so whether it is A is added, D is added, which nucleotide is added, you will get it. Okay, and this is the portable. This is very small size. I think इतना ही होगा आपका nanopore का size. and by the time you are planning to travel from Delhi to Hyderabad, you will get the sequence. That is that much fast. Kuch sequencing platforms and it takes 5 or 6 days. In some platforms, you can complete your you can get your data in 1 day, but there are really some systems where it will be completed in 2 to 3 hours. Okay, so this is the first portable mini ions. Other than this, there are 2 other variants, grid ions and chromatones. I've just kept these 2. Yes, and then if I say the nature of this nanopores, so there are different kind of nanopores which can be utilized for the sequencing purpose. Some biological nanopores like जैसे proteins are there, okay, which are naturally they are in nanometer size, so they can be utilized. Other than that, we use artificial things, okay. Hybrid nanopores are there, and the synthetic nanopores are there, okay. So these are the different examples, and these are the important points which need to be considered before taking any genome sequencing projects. Okay, so if I say first choice of sample. So how this choice of sample is important? For example, if you are taking samples which you have stored 2 years back in -80, okay, and the same plant you just grew and then you took the tissue. So what will be the difference? The quality of your extracted material will be good in fresh tissue, okay, other than stored one. Then the second important point is choice of method. Which method you are using, whether you are using any kit or if you are using any methods, and what buffer you are using, what is the pH of your buffer. The small things are very important, you have to take care of. Okay, and then quality requirements. आपने DNA extract कर लिया, but आपका quality for example अगर recommended 1.8 to 2 होना चाहिए and you are getting 1.5, आपने धीरे से supervisor को बिना बताए भेज दिया, तो क्या होगा? So and then choice of suitable sequencing platform. Okay. So once you are done the sequencing, what you get? Sequencing हो गया, it is done. Okay. Now what next? अभी आप काम शुरू हो रहा है. Yes. So you have to check the quality. Okay, how will you check the quality? So whether you got the sequences, I mean from nanopore or PathBio or Illumina, कहीं से भी आपको मिला, ultimately the file format which you got is FASTQ. ठीक है, आपको raw read मिल गया, अभी आप कहानी आगे शुरू है ना? How to analyze or how to do the downstream analysis. Okay. So there are some pre-sequencing quality check and post-sequencing quality check. So what is in our hand is pre-sequencing. Sequencing के बाद जो आपका friend होगा, bioinformatician होगा, जो collaborator होगा, he will perform. But this for this you will be responsible. ठीक है, आपका supervisor भी आपको ही डांटेगा, है ना? कि I mean sample quality control में it depends how you are handling your tissue or the material. आप उसको आइस में लेके आ रहे हो कि मैंने सेटी में लेके आ रहे हो, है ना? आपने प्रॉपर ग्लास यूज़ किया, आपके पे फिर ऐसे ही हैंड्स हैं, आपने जैसे भी करके कैजुअली यू हैव एक्सट्रैक्टेड टिश्यू. ठीक है, एंड देन आफ्टर आइसोलेटिंग योर मटेरियल, इनपुट मटेरियल, हैव यू चेक प्रॉपर? आपने फॉर एग्जांपल इफ़ यू चेक द क्वालिटी ऑन स्पेक्ट्रोफोटोमीटर, हो सकता है आपका स्पेक्ट्रो ख़राब हो, फिर क्या करोगे? इसलिए इट इज़ ऑलवेज गुड to have reading from 2 or 3 different instruments, okay, before planning for these kind of things. आप उसको जेल में भी चेक करो, okay. So these things you can do, and after getting data there are so many things. So first you have to do is fast QC. आपको पूरा adapters whatever you have ligated there, barcoding करा है आपने, those things you have to remove, okay, and then you have to perform the downstream analysis. So what happens in fast QC? So जो पहला काम करते हैं, फर्स्ट आफ्टर गेटिंग द डेटा, आपको रॉ रीड मिल गया, वी व्हाट वी डू, फास्ट QC हम चला देते हैं, ठीक है, क्वालिटी चेक, फर्स्ट क्वालिटी चेक. So these are the basic parameters. Okay, so this green tick means these parameters are okay. Orange means कि मतलब चल सकता है बट यू शुड इम्प्रूव इट, और रेड वाला तो मतलब बिल्कुल नहीं चलेगा, यू हैव टू वर्क ऑन इट. ओके, हाउ विल डू इट? बायोइन्फॉर्मेटिक्स यूज करोगे आप टूल्स. ओके, सो इफ in summary you are getting all the parameters in green mark, green tick, so it means your QC is pass. You can now do the latter things, I mean आपका assembly, annotation or downstream analysis, pathway analysis, whatever you have planned for. And if situations are like this, then you have to work on it. Okay, so this is the basic pipeline. You can find more. So we'll start with the tissue, then you have to take care of the protocols for isolation of DNA or RNA, and then after getting the raw reads, what kind of tools you are applying. Okay, so then you got the filter read. Now there is, if you are planning for de novo sequencing or reference based sequencing. What is de novo sequencing? De novo क्या है? Fresh, without no reference. और कोई बताएगा? What is de novo sequencing? De novo means when you are doing for the first time or you are doing from the scratch. Okay, कोई भी आपको as a reference कुछ है नहीं आपके पास, ठीक है. So there are different softwares for de novo and reference based mapping. Okay, and then once you make the scaffolds or assemble the genome, we do annotations, and annotations के बाद हम सारा केग एनालिसिस करेंगे, जीन कौन से पाथवे में है, बायोलॉजिकल प्रोसेसेस, मॉलिकुलर प्रोसेसेस, so many things we do. Okay, and then आपका वो you can visualize in form of heat map or anything you can make. Okay, so these are the application part. If you see NGS, you can do RNA sequencing, exome, metagenome. You can do the particular, if you are interested in organelle genome sequencing. Okay, epigenome study you can do, whole genome sequencing or resequencing, what you are planning you can do. So this next generation sequencing looks very fancy, but actually it is very challenging. So प्रॉब्लम इज़ नॉट प्रोड्यूसिंग डेटा. आजकल तो कोई भी टिश्यू ले करके दे रहा है ना, कंपनी वाले को हमारा RNA सीक्वेंसिंग करके दे दो, ठीक है, बट द प्रॉब्लम विद द इंटरप्रिटेशन आफ्टर गेटिंग डेटा, वो तो कुछ भी, अगर फॉर एग्जांपल इफ़ यू आर गिविंग सैम्पल्स, प्लांट का सैंपल का आपने दिया, आपको उसने फंजाई का दे दिया या फिर ह्यूमन सैंपल का डेटा दे दिया, आपको कैसे पता चलेगा? ओके, सो फॉर दैट यू नीड collaborations. Either you should be master in bioinformatics or you should have some friend which is good in bioinformatics. Okay, so you cannot do these all the NGS things if you don't have the proper infrastructures or the workstations. Okay, so all these things are required. If you have any questions you can ask. I'm not sure how much you got, but really. अब इसको क्लोज कर दो. I'll be happy to answer if any question is there. Otherwise, I'll ask question. सर लंच के लिए कहाँ जाते हैं लोग? ओके,
Now it's raining. It'll be here for few more minutes.
yes, any question? to do the transcriptome at different developmental stages. 5 days old plant, 10 days old plant, 20 days plant. आपने सारा लाइब्रेरी एक साथ अगर pull कर दोगे आप without any identifier, how you will identify कि 5 दिन वाला काम या 10 days वाला काम है? So that will be the barcode to identify your sample.
What will we use to tag this sample?
Sorry?
What will we use?
Tags identifiers they are जैसे ओलिगोस है ना उसी के फॉर्म में आपको वो मिलता है.
Like you have told about the ring values, right?
हाँ.
Like sometimes we will be taking samples in replicates. Okay, for every sample we will be taking in replicates. Like if in one or two replicates ring value is not good, like will it affect our output?
See, for all the samples, green value should be. I'm getting nowadays required one is about 8 or 9, but it should be at least that. And for example, if I'm having 3 samples, R1, R2, R3 for the same group, R1, R2 you are getting 8, and R3 you are getting 6.5. So you will be comment you will be getting like suboptimal. Suboptimal means कि अगर नहीं निकल पा रहा है तो चल सकता है because you are already having optimal. But it is always good that you should follow the What are adapters? Adapters क्या है? Why we are adapters? Now it's my turn.
Okay, thank you. Actually, this kind of lectures are arranged to give you the basics. Because as Dr. Sanjay Sharma said, anybody can outsource by giving the sequencing, whether they do the single sequencing or NGS. It's always preferable you should know the basics. What for we are doing is, only with understanding some basics if you do, then you can give the reliable interpretation, you can get it from the outsourcing one. Whichever you give for outsourcing company, they will give some data, but you should have the discretion whether what the data they are providing is good or not. For which you have to have the support of a genomic analyst specialist, you have to have in your team. That's more important. Just you have the paisa for 30 or 40%, you can give a transcriptomic data. For transcriptomic, you have to take it all consideration when the sample has to be collected. What is the quality of that? What is the real value of them? If you know the basics, reliably you can get the output. Once your input is not good, how much ever you spend, you will not get the reliable output. So that's why we import the training, some of the basics of this. Because with this one lecture, it is very difficult. Students are undergoing a whole course undergo on these aspects. What is the course title, madam? The course related to the sequencing you are offering at the biotechnology.
Advances in molecular techniques.
Advances 2+1 course, isn't it?
No sir, 0+2.
0+2 course is that for a whole course, for a whole semester,
She
they
has a
will be.
grade, sir. I think
No,
you
no,
have
for a
attended.
whole semester, 0+2, they will import techniques. So, I'm also on the way, this one class is not enough. Just it's like a curtain raiser. What is that? Just to give a panoramic view, it gives. For those who are interested in going there, you have to read a lot, undergo specific courses, then you can strengthen your repertoire of this genome sequencing technique. Okay? On behalf of the organizer, I give a small token of memento to Sanjay Sharma for delivering this lecture and for clarifying. Thank you, Sanjay. Nice. Now, as almost day is also over, just stop. You can go for lunch. You can assemble there at 2:15 at Discovery Center.
So if sir, anybody can further understand the basics, they can come to me. I will just send you my copy, pen and then
Yeah.
understand.
One more announcement.
So
you want any specific clarification,
We
you
can
can
come to
contact Dr. Sandhya Sharma at NAPB there on your off time. Okay, on a spare days, seeking your availability, you can contact her. Okay, Sandhya Sharma is accessible at the NAPB center there. One time we'll also have a visit there for their sequencing facility as a part of the training. Yes. Thank you. Thanks, Afi. Once again.
क्या सर के साथ? अच्छा.
बैग के साथ में